We can see your presentation. If you could just make a
Can you
presentation.
hear me?
Yes. Yes, now it's perfect.
Okay, great. So hello, everyone. Thank you so much for the opportunity to present what we're working on. So my name is Jose, and I'm the CEO and co-founder of Precise Health. And our vision is quite aligned with what Luis was talking about earlier, that phages will need an adaptive approach, and that we are also betting on the fact that phage therapy will be more realistic when we have this adaptive biologic dossier altogether. And so our mission is to help clinicians clinicians treat multi-drug resistant infections in a shorter amount of time than it is today, because what we believe in is that even when phage therapy will be the mainstream that everybody will be able to use them to treat patients, access to them will need to be fast enough. And that's built on the fact that majority of patients are actually that are suffering from AMR are within the acute subacute section, not the chronic cases which we are seeing today being used, where we are applying phage therapy. So what we've built is really the first global AI phage platform.
Last night, all right? Just
Sorry.
speaks really slow. This is what we want to encourage.
Please, I will unmute once.
Okay.
Everybody, please mute.
Okay.
I will mute again, everybody. Please mute. Okay, and now, Jose, you need to unmute yourself again. Sorry.
Well,
yes.
Okay, perfect. So yeah, I was saying that what we've built is the first global AI-driven phage platform, which is for clinicians and researchers. And what we're aiming to do is to connect diagnostics, data, and potentially therapy. And we want to turn what is currently slow and fragmented process into something that is actionable and scalable and potentially life-saving in the future. So given the complexity of the project and the problem that we are trying to solve, we have a multidisciplinary team, all based here in Switzerland, and we are combining multiple expertises, which is from microbiology, infection biology, bioinformatics, machine learning, and regulatory affairs altogether. And we are extremely convinced that our approach can help crack this phage economics and somehow manage to make it more scalable in the future if we focus on adaptive dossiers. So we all know that we cannot do that by ourselves, and nor should we do that. That's why we are very happy to be part of PhageEU to kind of have these collaborations open to us. So as everybody in the group, we are phage believers. And our journey to this belief started with one simple question that we posed to medical doctors, either using phages or have used phages in the past. And the question was, if phages can save lives, why aren't you using them more often, especially when all current treatments are failing in this case? And the answers are quite vast, but we have categorized them into five different, let's say, categories. And that's one. The first one is the production quality. What we saw today in compassionate use applications already is that the production quality, doctors have mixed feelings about this, because some are pro full GMP, some are pro GMP-like in this case, but the quality has to be clinical grade. So this is already quite good. And the reason for full GMP not being the big favorite in some cases is mostly because of costs that are implicated with obtaining that. And then the second case is the ethical hurdles, because I think this is something that is maybe some doctors say, okay, this is fine, because if I have a patient that needs the therapy today, we can overcome this ethical, let's say, approval in the board quite fast. But the three key things that were really a deterrent for them for using therapy today were, first, that for you to get the phage therapy, it takes just too long, because each time they want to use it, they need to go through so many hoops, which we can discuss in a little bit. And then this makes it even worse, because more than 80% of the infections that they see, they express that are within this acute and subacute category. And then when they want to use phage therapy and they need to access the phages from a specific phage bank, because most hospitals don't have phage banks themselves, right? So which phage bank should I send my phage to, my bacteria isolate to? How many of those should I send my bacteria isolate to? So this becomes quite of an issue. So in a nutshell, clinicians' key pain points are this delayed and complex access to fighting phages. And we think that as long as these hurdles are not solved, even if we have approval of phage therapy, regardless of whether it's cocktails or adaptive phage banks, it will still kind of have it will impair the scalability of phage therapy altogether. So today, finding the right phage therapy, you have to go through so many hoops. And I think some of you in this group already familiar with this, that you isolate the bacteria, send it to one phage bank or multiple phage banks. Potentially you search, manually screen, I don't know how many, tens or hundreds sometimes. We have heard about this from some doctors. And you find a match. If you don't find a match, you isolate a new one. If you don't find that, then you send it to another phage bank. And it's a whole loop altogether. So that takes a lot. And once you find it, then there is still the legal burden of it. How do I transport it from one country to another? Which MTA? Do we have a Nagoya protocol? All of these questions still are kind of unanswered today. So we have heard cases from the doctors that we talked to, and also researchers as well, that it can take 2 to 6 months to find these phages that they want to use. So with this in mind, we believe that fixing this gap between the diagnosis and potentially treatment in the future, once phages are actually available for mainstream use, is still necessary. So what we're doing at Precise Health is replacing these manual processes with an AI-powered, let's say, digital platform, where we are connecting an AI-powered digital phagogram, which is what we're developing as the Phage Match, and a simple sourcing platform as well. So here, the Phage Match, which is the matching platform, is the AI engine that helps clinicians and researchers digitally predict and source natural phages that will kill a pathogen with more than 80% precision. So these are our current metrics for some of the pathogens. And we want to connect a global phage bank network. So you would be able as a phage bank to list your phages into this platform, which would allow researchers and clinicians potentially to find them. And you can do all of this in just a few minutes, which I can show later on. And in addition to that, we are integrating and automating all the paperwork, essentially. So all the legal paperwork, including material transfer agreements, logistics, would be standardized and integrated and streamlined into this same platform. So it makes everything much easier for you to kind of transfer these phages from one place to another. So what normally happens today in several weeks, we aim to kind of shrink that and do that in just a few minutes, so to speak. Now, this is in a nutshell, what we have. So you would think of having multiple phage banks, which are unified into a single platform, where doctors could come, upload some diagnostic test of the specific bacteria. And from there, within minutes, you will be able to search into multiple phage banks, whether the phage is available or not. And depending on the quality, etc., that you would need as a researcher or as a medical doctor, you would potentially, in a few clicks, request that, and that will be sent to you. And this removes the burden where pathogens, I mean, already sending them from one country to another, it's quite a nightmare in terms of legal aspects as well, and also the logistics. So in this case, you don't have to send the pathogens anymore. You will just run a test in your lab and potentially get some matches in one side. So of course, we are starting with a research use application, and we want to potentially evolve into a CE-marked IVD, software as a medical device, which will serve as a clinical decision support tool for medical doctors in the future. And that would position us in the, say, phage therapy as it enters in the clinical practice altogether. So we get a lot of questions, of course, from phage banks, of course, because we talk to private and also the public ones. What do we gain from all of this? Why do we share our phages? Why would I share my information with you, and so on? And the reality is that we have seen is that, I mean, phages that stay in a phage bank somewhere and that are not being used. They're not helping anybody, right? So that's unfortunately the case. And you as a phage bank, in one way or another, either you want to share that with other researchers, or potentially your phage could help a patient that could be treated using your phages. So why not kind of have this shared, let's say, infrastructure? And we have been reached by many researchers who are willing to do this sharing infrastructure altogether. And we are happy to take, some collaborations on that. But I mean, of course, we can discuss about that later on in the discussion. And we were also asked to present a bit more on the scientific side, how we potentially train our models. And I mean, this is not rocket science. It's quite well published in many different publications. There are many different models, but the ones that have worked well for us are actually divided in 5 different steps. So the first one, of course, is the data generation, where we take all bacteria and phages, we run all these in vitro bacterial lysis, and we perform whole genome sequencing for all the phages, and then we prepare an interaction matrix for all of them. And then really the next step is to automate the bioinformatics annotation and data analysis. So we have automated all the short read, so you can start from FASTQ files. We have quality control, completeness assessment, etc. And we do all the genome annotation for bacteria and the phages. And from there, we identify certain parameters which we think might be linked to phage matching in the future. And of course, this is not an exhaustive list. We have a huge list of that. And once this is all done, what we do is to take all this information, including the matrix that we have generated during the data generation side, plus the metadata of each phage and bacteria that we have, and we train the models. And then we have a probabilistic output. And with that probabilistic output, what we have is a ranking of the confidence, based on the confidence level of how likely a specific phage is going to be lysing a specific bacteria, right? And there we have a cutoff of 80%. So I can quickly just go to this slide here. And as you can see from this, I don't know if my pointer is working. Oops, sorry. Basically, once we've done this probabilistic approach for each bacteria, we would have a ranking mechanism where we go from a low confidence to a high confidence level. And then we have a working threshold, which is 80%. And then for anything above this, then we are kind of already confident that we are able to say something, whether it's going to be lytic or not. And we have what we call the heat at 1 and 2 and 3. So what this is saying is that if I'm taking anything above this threshold of 80% and I say the top 3 phages, how likely to find at least one of them that works. And when we select 3, which is heated 3, we see that nearly 97% of the times we have at least one phage that works in the top 3. And if we do that for the top 2, essentially we have nearly 94% of the cases where we have the right phages. So how is this useful to anybody today? For the phage banks, of course, you received requests that, you need to test, I don't know how many phages when a patient needs a specific phage. Instead of testing hundreds of phages, you would just test, for instance, the top 3 in this case, because we know that most likely of those top 3, you would have something that would be working. And this could also be applied for GMP phages, non-GMP phages, regardless of the application that you want. So that is the direction that we are kind of going to. And of course, this in the future, currently we are working with whole genomes, etc., but we are also developing our own test that is quite fast, that you can perform in 2 hours. and we'll be able to match the phages from anywhere, including your own phage bank as a hospital in the future, without having to ship bacteria and performing like cumbersome tests altogether. Yeah, we also have a lot of doctors who say, oh, yes, you are matching phages, you're testing them in vitro, but how likely, I mean, what if you're giving me phages that are not safe enough? So, of course, this is why we perform some of the tests in animals, so just to validate, and we did the validation with multiple, let's say, bacteria and phages, also in Galeria and mice. But this is only to show that whatever is being recommended, which is in the pipeline, has some safety and efficacy that is required for in vivo studies altogether. So yeah, this is mostly that. I don't know how much time I have. I can also just briefly show.
You still have some time, so.
Okay, great. So I can quickly show just how simple we aim to okay, so sharing here. So can you see my screen? Okay, great. So this is the version 0 of the platform as it is. That, I mean, it will be open to anyone, researchers or clinicians who are looking for phages, but they want to be able to narrow down the number of searches that they would need to do. And essentially, this is the panel where you would have the phage match, the phage tailor, which is more on an advanced model for engineering, but that's something for the future. And then here you will have a phage database. And then for the phage database, essentially, we would allow any phage owner who has some phages which have been genomically characterized and with some metadata to add their phages in here that they could be searchable by anybody. And essentially what we have is that for each different phage, we would have all the metadata, including where it was used before, whether it was used in just in vitro or even in treating specific patients and so on. So you could list that and we would have all of this in here. And then in the second stage, of course, when you're searching for the phages, so instead of all these hoops that I was showing before, you would be able to just upload here a FASTA file at this stage. That's what we are working with. But we are also working on a diagnostic test, as I mentioned, which is much faster. So you could just upload the characterization of the bacteria into the platform. And what this will do is to run the analysis. And for each bacteria that you have run, you will be able to see at what level of matching score and coverage you are able to get for your phages. And in this case, for instance, the matching score is not great. Obviously, you don't want to be able to use that. But in some cases where we may, sorry, not this one, but maybe this. So here you have a high score, for instance, and then from here you will be able to select and put that in a basket and essentially have it requested to the phage bank to send it to you. And the phage bank, they will receive a notification that XYZ institution requested your phages, and you would be able to prepare them, and all the shipments and the documentation and MTAs and so on. It's all integrated into the platform, so you don't have to do all this work all over again. And yeah, you can confidently share your phages either with a price or for free, however you prefer. So that will be able. So that's how we are trying to move the needle. And I'm happy to take some questions.
Perfect. Thank you, Jose, for this very indeed interesting and promising tool. Again, a lot of congratulations. You said it's not a rocket science. I have to admit it is rocket science. So happy to share with you some questions that have been asked. And if you could help me indeed go through them.
Yeah, sure.
And that would be very, very useful. Just a moment.
Sure.
I will try to share them. Okay. So the first question, I can address it. How many clinicians did you ask? You have indeed presented the challenges that clinicians are facing, or why, for example, they are not so eager using phages. So how many clinicians have you consulted in your question?
So in our case now, we talk to around 11 clinicians. So there aren't so many clinicians treating patients with phages. So I think already this number is quite good. And yeah, I think that's the number there. But of course, if you know some more, we are happy to talk to them as well to understand more.
Today in the call, we have 160 people. So if you indeed want to respond to these questions, please feel free.
Yeah, sure.
To write your replies down.
Yeah.
And second question, who will do the bacteria sequencing? Do you offer it as a service, or you rely on other service providers for that?
So for the training of the model, we do our sequencing ourselves. So that's fine. And then for the doctors who would like to access phage therapy, I mean, we can help you do the sequencing because we also have our own sequencer in the lab. But of course, the idea is that you don't have to go through these logistics of, having to send samples from one place to another. So if you can take care of that in your own country or in your own location, I think it's much easier, because for us, all we want is that you upload the genomic information of the bacteria, and then we try to help you narrow down so that you don't have to, go through all these logistics, so to speak. But like I said, sequencing is just the start, because this is how we learn which features are actually implicated with matching and not matching. So based on that, I mean, we develop a test that you can run in a few minutes, so you will not have to do sequencing. I think by mid 2026, we will be launching that product soon.
Question number 3, is it for you? How working threshold 0.8 was estimated, based on what?
Yeah, good point. So we took multiple thresholds. So the confidence is given by the machine learning. So the model itself, how confident it is to, how do you say, give the right results, essentially. And then we took multiple thresholds. We started with 50, 60, et cetera. And then we did a confusion matrix on these different thresholds. And the best one that we found to be working well, or we have the best results, is the 80%. So what that means is that if you give us a bacteria into the platform, and for some reason, all the phages that are matching are below 80%, we will say, we don't want to recommend anything for this bacteria because we are not confident enough. But if it's above 80%, then we are confident enough. So that's how we came up with 80% working threshold.
Great. If you could take for me the question number 4.
Yeah.
A foreign.
Yes.
But indeed, it's about how you also deal with some errors resulting from contamination of therapeutic phages.
So how do we deal with the phage specificity testing errors resulting from contamination and therapeutic phage preparations with temperate phages induced from lysogens for propagation? And how do you only non-lysogens? So we only look at lytic phages for the start. We are not adding any lysogenic phages. And then for the phage specificity testing errors resulting from contamination, so for all the phages that we use for our training, we actually we do 3 rounds of purification before we even include that into the training model. So we minimize as much as possible. There will be errors for sure, but that's the best we can do, at least at this moment, I'll say so. I don't know if that answers your question, but yeah. Maybe for the phages that are coming from others, of course, we have a very good pipeline, bioinformatics pipeline, that is putting together from FASTQ all the way to, the genome annotations and so on and so forth. So we have some failed switch checks, safety checks for each one of them to make sure that it's also not lysogenic.
Okay, I'm not sure question 5 is addressed to you or maybe to the previous presentation. Can we?
Depending on this one.
The authorities that testing phage.
Innocuity is not necessary anymore based on the history saved profiles. I hope so, but I don't know if I can answer that correctly.
Yeah, as PhageEU, we can always try to
Yeah.
convince the authorities and decision makers. And the last question for the time being, a great demo. I suppose you have an interaction matrix for a subset of phages and bacterial hosts that you use for training, or all of them?
Yeah, so our main focus is escape plus E. coli, and we want to start with gram negatives, more specifically because the genomic diversity in this we have seen that is insane compared to gram positives. So we are not starting with a baseline of where we train with the whole forest, but we go species by species. So the model that I showed you was for E. coli, and we're evolving that to Klebsiella and potentially Pseudomonas and so on and so forth.
Okay, let's maybe take those two last questions.
Yeah.
What do you think about using AI? Yeah, I understand.
Yeah. Yes.
Models to build in silico phages. If you already heard about that, what do you think about models' robustness? How
Yeah.
to predict viral genetic sequences, knowing that viral sequences are highly exposed to evolutions?
What do I think about AI models? I love them because that's what we're doing. And of course, our goal with this is to build something that is robust enough that we could potentially in the future be able to replace the in vitro testing, so to speak, or at least to some degree be able to do that. So in terms of robustness, I think robustness can mean a lot of things. I always say that whatever model we develop, we have always to treat it with caution, because the robustness for me, it's number of data points that you use, but the number of data points which are diverse enough, right? So if we are building something which we just have data specifically from phages and bacteria from Switzerland, I cannot confidently claim that the same analytical performance I have here is going to be the same in Germany, for example. So unless I have these diverse data points, I cannot claim that it's robust enough for the globe, so to speak. And this is why we are always open to these collaborations from multiple places, and we have people sending us phages and bacteria so that we are also building something as robust, so to speak. And then how to predict viral genetic sequences. Knowing that viral sequences are highly exposed to evolutions. So this is a cumbersome work. We have automated as much as possible. We are not, how can I say, we are integrating various technologies into the same platform, but we need to learn from other people who are already doing this. So we will be looking at open source, let's say, prediction models and trying to implement that as we go. So the evolution is part of what we are also checking there. I don't know if that answers well the question, if I understood correctly as well.
I suggest then indeed maybe continue questions exchanges in the chat. I think question number 8, as well as those incoming questions, are a bit more specific, practical, how to use your platform. So please feel free to respond to them in the chat, because we are running a bit out of time, and I will now indeed invite our third speaker, Ville Hoikala, to take the floor.